Welcome back, my dear students. Today we are going to talk about embryogeny. First of all, what is the definition of embryogeny? It is the sum total of all the changes that occur during the development of mature embryo from the zygote. हर एक change that takes place, any change that takes place during the development of mature embryo from zygote, all those changes, all those steps that constitute the embryogeny. Okay, sum total of all the changes that occur during the development of embryo, mature embryo, you can say, from the zygote, all these changes they constitute the embryogeny. Now आज हम बात कर रहे हैं एम्ब्रियोजनी ऑफ डाइकॉट्स. जो डाइकॉट्स में एम्ब्रियोज होते हैं, the embryogeny in dicots has primarily been studied in the crucifers. Crucifers is a group of plants. Okay, they have been studied with respect to embryogeny. That is why dicot embryos, they are also known as crucifer type embryos. The crucifers, they are the representatives of dicot embryos. They represent the they have been studied extensively to study embryogeny in dicots. That's why dicot embryos, they are also known as crucifer type embryos. Now embryogeny or development of embryo in dicots, it takes place in a number of steps. You have to remember all those steps. The first one is you are having a zygote. Okay, ये embryo sac है. Here you have zygote. This zygote, it elongates along the micropylar chalazal axis. This is the micropylar end and this is chalazal end. The zygote, it elongate हो जाता है. Okay, it then divides by the first mitotic division, which is asymmetrical, or you can say uneven, and this division is transverse, producing a small apical cell or terminal cell or embryonal cell towards the chalazal end, and a large basal cell or suspensor cell towards the micropyle. This is zygote which has elongated. It divides into a small apical cell. This is small apical cell towards the micropylar end, and large basal cell towards the this is towards the chalaza, and this one is towards the micropyle. Okay, this large basal cell, which is located towards the micropylar end of the embryo sac, it divides transversely a few times and produces a filamentous suspensor of 6 to 8 cells. Now you are having small apical cell, and this large suspensor cell, it divides few times and results in the formation of filament like structure which is known as suspensor, and this suspensor is constituted of 6 to 10 cells. जो लास्ट वाली सेल होती है सस्पेंसर की, उसको हम बोलते हैं हास्टोरियम. ओके, तो याद रखा आपने, जाइगोट डिवाइड होता है, फॉर्म्स 2 सेल्स, स्मॉल एपिकल सेल एंड लार्ज बेसल सेल. जो बेसल सेल होती है, इट डिवाइड्स फ्यू टाइम्स, resulting in the formation of a suspensor of 6 to 10 cells. Okay, now the suspensor, the function of suspensor is it helps in pushing the embryo into the endosperm. जो suspensor cell होती है towards micropyle, it pushes the embryo, developing embryo, deep into the endosperm. Okay, जो endosperm food होता है बीच में, उसी में push करता है ये suspensor embryo को. The first cell of suspensor towards the micropyle, micropylar end, sorry, it becomes swollen and functions as haustorium. जैसे कि मैंने आपको बोला, this is the apical cell, then we have a few celled suspensor, 6 to 10 cells usually. This cell, the cell of the suspensor towards the micropylar end, it becomes swollen and functions as haustorium. It functions as histurium. It pushes the embryo, developing embryo जो इस वाली apical cell से बनती है, it pushes the developing embryo into the endosperm. The last cell of the suspensor at the end adjacent to the embryo is called hypophysis. ये वाली cell जो embryo के साथ attached है इसको हम बोलते हैं hypophysis. Hypophysis later on gives rise to radicle and root cap. याद रखना, this is very much important. जो hypophysis है, that cell of the suspensor which is adjacent to the developing embryo, this is known as hypophysis. Later on in during the course of development, it will form radicle and root cap of the embryo. The embryo, the embryonal cell, or you can say apical cell, it undergoes two vertical divisions at right angles to each other to give rise to four cells. अब बारी आती है एपिकल सेल की. ओके, बेसल सेल का हमने देखा, वो सस्पेंसर बनाती है. अब एपिकल सेल की बारी आती है. हियर वी हैव दिस टाइप ऑफ स्ट्रक्चर. अब ये एपिकल सेल इट विल डिवाइड by two walls which are vertical, okay, which are vertical and are placed at right angles to each other. दूसरा डिवीजन भी हो जाएगा इसमें. ओके, फॉर एग्जांपल ये एपिकल सेल है, एक डिवीजन ऐसे हो जाएगा, दूसरा डिवीजन ऐसे हो जाएगा. हाउ मेनी सेल्स विल बी फॉर्म? 4 cells. ओके, एंड दिस स्टेज ऑफ एम्ब्रियो इज कॉल्ड क्वाड्रेंट स्टेज. यहाँ पे इसके पीछे और एक सेल है, ये एक सेल है, इसके पीछे और एक सेल है. उस ठीक है, ये एक सेल है, इसके पीछे और एक सेल है. सो द एपिकल सेल it undergoes two vertical divisions at right angles to each other to form 4 cells, and this stage of embryo is called a quadrant stage. Now the quadrant, it undergoes transverse division to form 8 cells arranged in 2 tiers. The outer tier, which is also known as epibasal tier, and suspensor tier, which is also known as hypobasal tier. याद रखना, जो आपके पास क्वाड्रेंट है, जो एपिकल सेल थी, स्मॉल एपिकल सेल, उसमें 2 डिविजन्स हुए and it resulted in the formation of 4 cells known as quadrant stage. Now जो quadrant में 4 cells है, all these 4 cells they will divide transversely to form 8 cells which are arranged in 2 tiers: outer tier, which is also known as epibasal tier, and suspensor tier, which is also known as hypobasal tier. And this stage in the development of embryo is called octant stage. यहाँ पे आप देख लीजिए, this is आपके पास 4 cells थी. ओके, ये सस्पेंसर है, ये हमें पता ही है. तो ये हम ऐसे देखेंगे, ऐसे था. ओके, क्वाड्रेंट का स्ट्रक्चर कुछ ऐसे था. अब क्या है, जो ये 4 cells है क्वाड्रेंट की, दे विल डिवाइड लाइक दिस. ये 2 cells थी, ये एक सेल. ओके, ये 2 थी, अब ये 4 बन गई. ये भी 2 थी, अब ये 4 बन गई. सो इट रिजल्टेड इन द फॉर्मेशन ऑफ 2 tiers of cells. This is known as epibasal tier or outer tier, and this one is known as hypobasal tier or suspensor tier. याद रखना, इस वाले टीयर में 4 cells है, 2 ये है जो हमें दिख रही है और 2 इसके पीछे है. इसी तरह इस वाले टीयर में 4 cells है, 2 हमें दिख रही है और 2 इसके पीछे है. ओके, सो दी quadrant, it divides transversely to form 8 cells which are arranged in 2 tiers: outer tier, which is also known as epibasal tier, and suspensor tier, which is also known as hypobasal tier. And this stage in the life in the development of embryo is called octant stage. Now what is the fate of epibasal and hypobasal tears? जो epibasal tear होता है, it forms two cotyledons and plumule, or you can say future shoot of the plant. याद रखना. The outer tear or epibasal tear, it forms two cotyledons and the plumule, and hypobasal tear or the suspensor tear, it produces hypocotyle except the tip. याद रखना, it produces hypocotyle except the tip, because the tip is produced by hypophysis, जो हमने पहले ही डिस्कस किया हुआ है. ओके, तो याद रख सकते हैं आप, जो एपिबेसल टीयर है, it forms two cotyledons and plumule or future shoot, while as hypobasal tear produces hypocotyle except the tip. Now you are having the 8-celled embryo, pro-embryo, sorry. Okay, you are having the 8-cell structure, which is known as octant. Now the 8 embryonic cells or the octant, it divides periclinally to produce an outer layer of protoderm or dermatogen and an inner layer. So आपके पास अब 8 cells थी, इस तरह थी कुछ. ओके, और हम ऐसे भी दिखा सकते हैं. एट सेल, ओके, 4 हमें दिख रही है, 4 नहीं दिख रही है. एंड दीज 8 cells, they will divide like this periclinally, parallel to the surface. Okay, they will divide periclinally. तो क्या हो जाएगा, 2 लेयर्स बन जाएंगे. अंदर वाला लेयर composed of 8 cells and an outer layer composed of 8 cells. जो बाहर वाला लेयर होता है, इसको हम बोलते हैं प्रोटोडर्म और डर्मेटोजन. याद रखना. So how many cells you are having at this point of time? 16 cells. 8 constitute the protoderm or dermatogen, and 8 inner cells, they will divide further. The inner layer of cells, they divide further into procambium, which is also known as plerome, and ground meristem, which is also known as peribellum. याद रखना, this is very much important. The 8 embryonic cells or octant, they divide periclinally, producing an outer layer of protoderm or dermatogen and an inner layer. The inner layer, it divides further to produce procambium and ground meristem or peribellum. And at this stage, the embryo is called globular pro-embryo. The embryo at this stage is called globular pro-embryo. The development of various organs of the embryo occurs in the pro-embryo stage. याद रखना, this is very much important. I will repeat this. Now you are having this type of structure. You have an octant stage here. All the cells of the octant, they will divide periclinally. तो अब आपके पास 16 cells आ जाएगी. Outer 8 cells and inner 8 cells. The outer 8 cells, they are known as protoderm. They constitute the protoderm or dermatogen. And the inner layer, it divides further to form procambium or plerome and ground meristem or peribellum. Now at this stage of the embryo, all the organs of the embryo, they develop. Development of all the organs of the embryo occurs at this pro-embryo stage. और याद रखना आप, इस स्टेज को हम बोलते हैं globular pro-embryo stage. Now you know you are having an outermost layer of cells of the globular pro-embryo, which is called as dermatogen or protoderm. This dermatogen or protoderm, it gives rise to epidermis. Okay, याद रखना, you have now 3 layers. One is dermatogen, second one is procambium, and third one is ground meristem. Each of these 3 layers, they will give rise to specific organs. फॉर एग्जाम्पल डर्मेटोजन जो है या प्रोटोडर्म जिसको हम बोलते हैं, इट गिव्स राइज टू एपिडर्मिस, प्रोकैम्बियम और पेलिरोम गिव्स राइज टू स्टील. Steel means vascular cylinder, vascular tissue. Okay, procambium gives rise to steel, and ground meristem or peribellum it gives rise to cortex and pith. जो peribellum है it gives rise to cortex and pith. The two cotyledons they differentiate from the sides with a faint plumule in the center from this globular proembryo. आपके पास अभी globular proembryo है. Okay, the globular proembryo it forms two cotyledons. The two cotyledons they start differentiating at this stage with a faint plumule in the center. जो globular proembryo है, at this stage the two cotyledons they start differentiating, and at this stage you will find a small plumule in the center of these two cotyledons, and at this stage the embryo becomes heart shaped. Now the embryo becomes heart shaped. अभी ये globular था, अब ये heart shaped बन गया. Okay, after the differentiation of the two cotyledons the embryo has now assumed a heart shape. When the cotyledons are completely formed they enclose the plumule at the base, and when the cotyledons have finally matured, they have finally reached the full development, they enclose a plumule at the base. We can see this on the next slide. Here you can see how embryo development takes place in dicots. You have here zygote, it elongates, divides, forms a terminal cell or apical cell and basal cell. जो basal cell है, it forms the suspensor here, 6 to 10 cell suspensor. Now the apical cell, it divides and forms the quadrant. The quadrant divides and forms the octant. The octant divides and forms the globular proembryo. Now the globular proembryo here, it starts differentiating the cotyledons and it becomes heart shaped. Okay, the heart shaped proembryo it then finally develops further. The cotyledons they reach the full they become fully matured. The cotyledons become fully matured and they enclose a plumule at their base. Okay, or you can see this one. This is more clear. Here you are having the zygote. It divides, forms apical cell and lower basal cell. The basal cell it gives rise to suspensor that has not been shown here. Okay, the apical cell it divides by two transverse vertical walls at right angles to each other, resulting in the formation of quadrant. And this quadrant it divides transversely, forming the octant. Okay, the octant has not been shown here. The octant it divides all the cells of the octant they divide periclinally, resulting in the formation of 16 cell stage. The 16 cells here you can see they are in the form of two layers, outer layer which is known as dermatogen and an inner layer. The inner layer it divides further and gives rise to procambium and the ground meristem. The procambium forms the stele and the ground meristem gives rise to cortex and pith. Okay, now this is the early globular proembryo. This is the late globular proembryo, and in this late globular proembryo the cotyledons they start differentiating. Here you can see this yellow green patch of cells, they are destined to form the cotyledons. When the cotyledons become fully mature, they attain their full development, they enclose a group of cells here which later on form the plumule. Okay, they enclose a plumule between them, and finally when all the development has taken place, you have this embryo which is having an embryonal axis. Okay, and two cotyledons. The embryonal axis, it has the pilumule at the tip, which forms the future shoot. Then we have the hypocotyle, this one, okay, and at this point you have the radicle which gives rise to future shoot, future root, sorry. This is hypocotyle, this is pilumule, these are the cotyledons, and this is radicle. This radicle gives rise to roots, future roots, okay. So this is all about the embryogeny in dicots. I hope you understand all the steps. uh, for the sake of convenience, I have not gone into the details of each and every step. You can simply remember that the zygote, it gives rise to two cells, apical cell and basal cell. Basal cell forms suspensor. The apical cell, which is also known as embryonal cell, it undergoes a number of repeated divisions and results in the formation of quadrant, then octet, then 16 cell stage. Then this 16 cell stage, it gives rise to different organs of the embryo, okay. It gives rise to different organs of the embryo. First of all, uh, this early globular proembryo, okay, it gives rise to late globular proembryo. This late globular proembryo, it starts differentiating the two cotyledons here. When the cotyledons become fully mature, they enclose between them the pilumule, okay. So this is all about the embryogeny in dicots. See you soon with a new lesson. Till then, take care.